Welcome everybody, ongi etorri. Bienvenidos. This is all my Spanish and Basque that I have for you. But it's my pleasure on behalf of the organizing committee to welcome you all to Neurogune 2026. So this is the 7th edition. This thing is a little bit like, I don't know. Yes, 7th edition of this event. And it's the biggest one that we ever had, at least that they told me. I've never been here. But it seems that you guys are already having fun. I hope that everybody was able to find their poster in the end, their poster place, and hang it. If you still have problems, please come to us during the coffee break and we'll get it fixed. Some numbers, there's like 235 registrations, so that's a lot of people that all came from the Basque country and even abroad. Like I think there are people from the UK, from France, even from Turkey maybe. Oh, there's like more people coming, so I'll give them some time. Were you all able to hang your poster? Was it okay? Yes. Please take a seat. So, I think now everybody is here. So again, ongi etorri, welcome, bienvenidos. You know, we do as we do, right? So yes, welcome. As I was saying, Neurogune 2026, the 7th edition of this beautiful event. There are 235 of you, more or less, so that's a lot of people coming from the Basque country. And 60% is like pre-doc or master students, so that's a lot of early career scientists that means that, you know, Basque neuroscience is healthy, and we hope that we can foster some collaborations and interactions between you all, so that we can become even stronger and that more people will come in 2 years. And like what I wanted to say is also that, you know, I'm very happy to have you all here, and that I'm very honored that you invited me to be the president of this thing. So thank you all for this confidence in me and also in Monse. And so maybe you also heard from my accent, but I'm coming from the Netherlands. So I'm like a Dutch person, and the person that I wanted to introduce today is Roelof Hut. So I, of course, because I was the co-president, I was able to invite him. And what I think for neuroscience is very interesting is comparative neuroscience, right? So one of the things with neuroscience is that like maybe we focus a little bit too much on one species, which is the mus musculus. And maybe we should like use biology, variation of different species to see like how neural circuits regulate behavior, right? Because there are so many different species that have all these different solutions to their problems, to adapt to their ecology. And well, the work of Roelof is very exemplary in this field. He has worked with a lot of species, mostly mammals, but I think also birds and insects. And I think he's also venturing into the crab world. So that's very, very interesting. I don't know if you're going to talk about it today, but his work is mostly centered on chronobiology, so that's like biological rhythms and how they influence metabolism and cognition, and also reproduction. So yes, he has published in very influential papers like Science, PNAS, and the best one of all, Current Biology, which is like my favorite journal. And yes, I want to give him the chance to give his talk, which is called The Temporal Niche Arms Race, an evolutionary view on biological rhythms. Thank you. And thank you all for coming.
Thank you very much, Arjen, and Monse, of course, for inviting me. I hesitated actually to come, to be honest. Because my wife and I planned to have our holiday around this time. So I had to negotiate between Arjen and my wife whether I could come or not. But as it turns out, everything's fine. My wife said, why don't we go on a holiday to the Basque country? And I have to say, it didn't disappoint us at all. It was a wonderful reason to spend time. And I actually managed to have a scientific slide of this region in my talk that you'll see. So there's a little promise there. So what is the temporal niche? We are studying biological clocks. And biological clocks time our sleep, and it means that they also time when we are awake, amongst many other things. And this is really an adaptation to the planetary movement. I feel a little bit, there's no handheld microphone, so I have to sort of wiggle things a little bit. There's a pointer, I think, here. Yeah, let me see. Does this work? Things are always terrible. There is, yeah. So this is, by the way, let me go back one slide, I forgot something. So we wrote a book on circadian rhythms together with Bill Schwartz, who was a big name in the circadian field, in biological rhythms. And we recently wrote a book on the essential principles practice of circadian biology, and it's freely available. So if you go to opentextbooks.rug, University of Groningen.nl, rug.nl, circadian biology, you can actually download this book as a PDF and you can use it. So it's freely available. If you run any courses that has some circadian biology in it, then I think there are many original figures in there that you can use. So make use of that. So if you, this is one of the pictures from the book actually. It's the planetary movements. The Earth turns around its axis every 24 hours. And that creates a light-dark cycle in the environment. And from the early on, life has adopted adaptation to that light-dark cycle. But there are more movements here, of course, that we will not talk about in this talk. There's the orbit of the Earth around the Sun takes a year. And that generates, because of the tilted axis, a difference in day length. And actually, like Arjen, who is working on voles, we've been working on voles as well, and we studied the influence of this day length difference, which changes their whole physiology, not only the reproductive system, but also the growth rate, etc. So there's a huge impact on just the length of the light-dark cycle. And then there's the lunar cycle that generates tidal rhythms on Earth. And this is something we will study in the next project that we just got funded, and that's the crab work that we will do. So we will study sleep in crabs because nobody's done it so far. So there you go. Now let me try to, maybe now it works. So let's go to the early emergence of life. 4.5 billion years ago, Earth started to become in existence, and every turn of the spiral, I really love this slide, every turn of the spiral is 1 billion years. And first life already emerged 3.8 billion years ago, so it took a little bit longer than 1 billion years for life to emerge. Now that early life was bacteria, and here they are. You can find traces of that. Layered bacterial colonies, stromatolites. Those are really the earliest life forms that we know of. And they are cyanobacteria, and they were photosynthetic bacteria. And here they are, they look green. They still form these colonies you can find on the coast of Australia. And the fascinating thing is that they photosynthesize during daytime, of course, generate ATP. But how do they survive the night? And this is a fundamental problem. And these first life forms have already solved this by having a large molecule that has 12 ATP phosphorylation sites. So they actually, ATP can donate a phosphate molecule, and it phosphorylates these phosphorylation sites. And that works as a energy storage. And in fact, what Takao Kondo, who passed away a couple of years ago, has found that if you have this Chi-C molecule in vitro, in a glass vial, it actually oscillates by itself. There's a whole research groups have studied this process, so I'm cutting it very short. But it means that the very early life forms, these bacteria forming these colonies, already have a protein-based clock that can function as an energy storage device. It's pretty amazing. I'm just pointing out that that energy storage can be, or energy balance is essential. And I will talk about mammals during the rest of the talk, but again, keep in mind that energy balance is essential. So if we look at mammals, then they started to appear, vertebrates started to appear 360 million years ago in the oceans, and then they came on land, and the Archosauria started to sort of invade the land area, and then mammals appeared 200 million years ago, but that's way before the dinosaurs got extinct. So we had, as mammals, an overlapping period of, 146 million years, where we had to live together with the dinosaurs. And this is called the so-called nocturnal bottleneck, because the dinosaurs were cold-blooded, as it were, and they had to be active during daytime, taking advantage of the heat during daytime. When it's light by the sun, it's also warm. It's something you know here in Spain. It's pretty warm. When we went to the Navarra, it was bloody hot already. But the dinosaurs got, profit from that. And the mammals, in order to escape the predation of the dinosaurs, they had to stay in their burrows during daytime and be active at night. And so the first mammals were actually nocturnal mammals. And that all changed at the extinction of the dinosaurs when a meteorite, an asteroid hit the Earth, and that at the time of the KPG boundary. So that's here, that's the boundary of the Cretaceous between the Paleogene. So the KPG boundary is marks the end of the dinosaurs. But when we started studying this, we found that actually these dinosaurs already had some adaptations and became already nocturnal. Or had the smaller, small, about 2 meter high, Velociraptor, Oviraptor, etc., those smaller predatory dinosaurs, we know now that they had feathers. So if you're watching Jurassic Park movies, etc., then you know it's all bullshit, because actually these dinosaurs should have had feathers. And that evidence has accumulated over the last 10, 20 years. So it's these predatory dinosaurs that developed feathers that led to the birds, of course, but also that led to the fact that they became more insulated, and therefore these first feathers were not for flight, they were for thermal insulation. And so therefore they could evade the nocturnal niche, as it were, and start to predate the mammals. And that generated an arms race for what we call the temporal niche. The temporal niche as an ecological niche during the Cretaceous. And so the mammals and the dinosaurs showed some flexibility. And in fact, if you watch now more recent publications, this is 2017, but already was mentioned in 2012 by Mario das Reis, before the mammals start to radiate here, this is the mammalian descendant tree, and they start to radiate already way before the KPG boundary. So it is this nocturnality of the dinosaurs, that's what we propose in that paper, the nocturnality of the dinosaurs that sort of caused mammals to diversify in their ecological niche. And we see some diurnality here in yellow starting to appear, and then it becomes more massive after the dinosaurs went extinct. And we actually studied more, if you're interested in it, I'll not talk about this, but we said, okay, maybe these circadian adaptations can form this temporal niche. So not only the ecological niche is important for speciation and diversification, but maybe also this time organization, this temporal niche is important. And we started studying this in more detail in these two papers, and actually we found that circadian rhythms can cause speciation depending on whether they're under sexual selection or not. And I hope that this will be quite influential, because I think it may be one of the most important papers that we published. Now, during my trip, I promise you this picture. It's wonderful. If you go to Diba, here's my wife, by the way, who kindly allowed me to speak here. If you go to Diba, just east of here, you can see these layered rock formations that are all along the coast. But at Diba, you can see the remains of this KPG boundary. It's this little layer that you see here that has been identified as because of the deposits of this asteroid. And so here is the remains of that. And it's the first time for me that I actually see the evidence of the KPG boundary. So that's fantastic. So if you don't know this, then maybe you should visit that area. It's all really well explained as well. So the question now is, can we still see traces of this temporal niche flexibility in mammals today? And we devised something that we call the circadian thermoenergetic hypothesis. And you'll hear, we published quite a series of papers on it, and you'll hear pretty soon why we call it the circadian thermoenergetics hypothesis. But the idea is that mammals, they originally are nocturnal to evade predation, but this is causing some energetic problems. So the diversification of mammals led to a lot of different rhythms, and these are all animals that we have studied. Here is the diurnal crown squirrel, the most extreme diurnal mammal that is known. We described this with light loggers, and it is actually, you can see that the animal is above ground with the black bars here through the days of the month. And you can see that the animal actually never sees sunrise or sunset. It's so afraid of darkness, it sort of stays away. It also has a retina that consists of all cones, so it's completely adapted to daytime vision. Here we have a nocturnal adapted mammal, the house mouse. You can see that this is laboratory data. These were field data, but this is laboratory data. If you run them in a long night, then you see the activity is limited really at the night. But even if you compress the night to just a few hours, these house mice try to keep their activity in the night. So that's really a nocturnal adaptation. But we will see that we can change this mouse. If we play around with the energetic balance, we can change this mouse into a diurnal mouse. Then we have crepuscular animals, and these are typically hoofed animals, so the ungulates. All ungulates are basically crepuscular, means that they have an activity peak around dawn and around dusk. This is throughout the years for males and females, and these are red deer in the Netherlands that we studied, but we also studied them with GPS transmitters in Canada, and there's quite some interesting differences and responses to the nocturnal or diurnal predation that is happening. Won't talk more about that, but it clearly shows this plasticity. And then we have the ultradian rhythms of voles, so I just put this in because of Arjen's work, of course. And here you see that these voles, they seem to be neither nocturnal nor diurnal, but they are active throughout the day in rhythms that have, sleep-wake rhythms that have like 3 or 4 hours rhythmicity. And the amazing thing is that these rhythms are synchronized in populations in larger areas. And these family groups synchronize through huddling. And that's maybe something nobody knows, but maybe it's something that the oxytocin hormone is involved, who knows? But they need to huddle together, and that synchronizes these short-term sleep-wake rhythms. And also here we see that energy balance affects the expression of these rhythms. So the idea is, all the fundamental work is that mammals have a hypothalamus, and in the hypothalamus, this is based on mouse work, but also in humans it's clear, the hypothalamus has a suprachiasmatic nucleus here, and the suprachiasmatic nucleus just sees the light-dark cycle. Specialized photoreceptors in the retina, they project to the suprachiasmatic nucleus, and that is the major dominant circadian clock in the body. The clock genes have been found as BMAL, and clock is a heterodimer that forms a transcription factor complex that drives the period and cryptochrome genes, and they have a negative feedback on the BMAL and clock action. They just pull the BMAL and clock from the promoter sites, and then the transcription stops. And so there is a negative feedback that generates these oscillations. And if we put luciferase just at the PER gene, the PER2 gene in this, the PER2 gene in this case, we attach a luciferase reporter gene, we can actually look at the hypothalamus and visualize the suprachiasmatic nucleus, and put it in culture, and then we can see those neurons lighting up with a 24-hour rhythm. This is much faster because 1 second here, I condense this movie. 1 second is 24 hours in real time. And so we see these wonderful oscillations, and if you quantify them, they actually form a wonderful sine wave. And that is what we call the clock. Now, these transcription factors, they also drive many other output genes, TEF, HLF, but we call the whole family clock control genes, and that affects daily timing in the cell and in the physiology of the body. Now the question is, if the suprachiasmatic nucleus listens to the light-dark cycle, or sees the light-dark cycle, it's always entrained in such a way that light induces purr. So if there's light outside, purr is going up. And no matter what your activity rhythm is, whether you're nocturnal or diurnal, that's how it is. The SCN entrains to the light-dark cycle. And anything else, flexibility in activity or sleep-wake patterns, happens downstream of the SCN. And this is something that is a mystery of the field, and that we're trying to solve that in this talk. Well, we talk about the functional relevance of the circadian clock. Well, there's an external function, the synchronization of the light-dark cycle and the temperature cycles. But there's also an internal function that we'll talk about, and that is synchronization of peripheral tissues, because all the organs have their own clocks, and they need to be synchronized as well. So there's an external function relative to the outside world, but also an internal function of circadian organization. So here we have this SCN again, and we know that the SCN is synchronizing all these rhythms in the tissues. These are all luciferase traces, PER2 luciferase traces, and that body synchronizes them through pineal melatonin, but also glucocorticoid signaling from the adrenal gland is important. We'll talk more about that. And body temperature is important. Simple fluctuation in body temperature is synchronizing the circadian clocks in your peripheral cells. So temperature is important for the internal milieu of the body, but the outside temperature is also important. And here we have a study in the outdoor enclosures that we're using with Vincent van der Winne. If you have an ambient temperature pattern, and these are real data, that looks like this, it's high during the daytime, it climbs up during daytime because of the heat of the sun, and then it cools down, then in the burrow or in the nest, you see a dampened rhythm, but also you see that it's a little bit delayed. Okay, it's very, very clear. Now, a nocturnal animal will be outside when it's cold, and then it will see the lower part of the curve when it's hiding in the nest, and it'll see outside cold temperatures again. So a nocturnal animal will see the lower part of these combined curves, but a diurnal animal will see the upper part of these curves, if you think about it. So a diurnal mammal is exposed to higher temperatures 24 hours compared to a nocturnal mammal. And that actually saves about 10% energy if you calculate this for the mouse. So we said, okay, what's now happening? And this is a mouse in a running wheel. If we let a mouse run in a wheel and let the mouse work for food, and here you see a food dropper that drops a little pellet. The mouse needs to run, and we can increase, of course, the amount of revolutions the mouse needs to make for the food to drop down. And you see the mouse is listening. Did I hear a tick? Did the pellet drop? Ah, now it is. And it eats the pellet. And we can increase the workload to such an extent that the mouse is losing weight gradually. And you'll see that they eat every crumb of the food, and they learn how to work for the food. And this is a very, what we think, ecological situation, because the mouse needs to walk around and look for the food. And if we do this in voles, with Laura van Rosmalen did this, you see that the vole can be nocturnal predominantly, but then if we let the vole work for food and it's working so hard that it's on a negative energy balance, it's losing weight, you can see we really can titrate this over a period of about a month. Then we see that nocturnal activity turns into this beautiful ultradian rhythms. And we think that's an adaptation because these ultradian rhythms in the voles are the most efficient digestion system. You load the fermenter with little portions of food. And so that's probably an adaptation to have more effective energy balance. If you do this in house mice, you see a completely different pattern. Oh, by the way, the colors here indicate body temperature. And it's quite important because we see that the body temperature of these voles, they drop a little bit, but never below like 36 degrees. But if you do this in house mice, you see a completely different pattern. We see that these mice, and this was done by Violetta Pilots, you see that these mice, they actually become diurnal. We see that the activity pattern shifts due to the daytime. And we also see that they can show torpor here. The body temperature drops to about 20 degrees. It's pretty amazing. This is an adaptation that's completely natural to save energy. So the amazing thing is, of course, that if you keep in mind these temperature patterns, the highest temperatures are here now towards the end of the light phase in nature, where the mouse is active, and the lowest temperatures are here towards the dark phase, when the mouse is showing torpor. And that's the most optimal time to be active, but also the most optimal time to show torpor. So both are an adaptation, and both save about 10%, so 20% in total, if you do the calculations of the energy balance. So this shift to diurnality is an adaptation, we think. Now, well, if that's an adaptation, then maybe what's happening if you just lower the ambient temperature, and indeed, by just lowering the ambient temperature, you increase the amount of diurnal activity. There was an experiment that nobody's ever done, but if you quantify it, it's a beautiful increase in diurnal activity as well. If you increase the workload at different ambient temperatures, you see that the lower the ambient temperature, the earlier in the workload program these mice show diurnal activity. So this all fits this so-called circadian thermoenergetic hypothesis that animals can use their adaptation, their flexibility especially nocturnal animals can use their flexibility in the activity program to save energy. Now, if you look at in the brain, in the hypothalamus, we see that the SCN, as I predicted in the control ad libitum and the work for food, just shows the same pattern. Clock genes don't change. If you look at the liver, we see a shift in the liver. The peripheral organs can advance in these diurnal mice. If you look at the adrenal, we see a shift in the peak timing even more, and we see that corticosterone is elevated, but also advanced. And this is important. We'll talk more about corticosterone. So then together with my former PhD student, Laura van Rosmalen and Sachin Panda, the Salk Institute, we said, okay, what's going on here? And Sachin Panda is a great colleague. He says, do you have any ideas? And he said, well, no, but I know a way to find out. I said, well, what's that? He said, well, we sequence the hell out of them. So that's his answer to all problems in biology, I think. But we did this wonderful study, and as it turns out, we see that in these diurnal mice here in green, we see no difference in all the clock genes. So the clock genes in the SCN stay the same. But in the SCN, this is now we did a lot of tissues, but this is in the suprachiasmatic nucleus. We see an amazing response that we have a whole set of rhythmic genes, 582 rhythmic genes, in the nocturnal mouse. But those genes in the SCN become almost arrhythmic if the mouse becomes diurnal. And the amazing thing is, if we look at the diurnal mouse, we have about the same amount of genes that show a beautiful rhythmic pattern. But if you trace those genes in the nocturnal mouse, you see it fades away and it's almost not rhythmic anymore, not significantly. So it is as if these SCN neurons, they have a circadian clock, and those genes don't change. They're attached to the light-dark cycle, but they have a clock control package of about 600-700 genes, and that's a diurnal package or a nocturnal package of genes, which is pretty amazing. So the interesting thing is now, the question is, what are those genes, and is that really the program that goes on? Now, if we look at other brain regions, here is a whole list of them. Nothing much happens. Here is the SCN, but we see especially that jumps out, there's some peripheral tissues here, what jumps out that speaks to us is the habenula. Thousands of genes change in the habenula, and that was a surprise to us. And if we do the same plot for the habenula, we get the same picture, rhythmic set of genes, about 1,000 in the nocturnal mouse that become arrhythmic in the diurnal mouse, and a set of 768 genes that is significantly rhythmic in the diurnal mouse, and they become arrhythmic in the nocturnal mouse. So we see a very similar pattern in the habenula compared to the SCN. And so why the habenula? Now, maybe some of you know that habenula is part of what we call the epithalamus. And together with the pineal gland, that forms an old timing system that is relevant for reptiles. So it is almost as if we're looking at the timing system that was important for reptiles and perhaps for the dinosaurs at the time. And it's as if this mouse becomes diurnal. Well, we like to think that it's sort of adopting the old reptile timing system. It's recruiting this old latently present timing system. But of course, it requires much more details. Let me see, time-wise. Well, very briefly, we had a second paper out of these studies. We said, okay, if these mice work so hard, we found, we don't talk about male-female difference in this paper, but we did find that, of course, as we all know, that the female mice tend to work much harder for their food than the male mice. Male mice are lazy mice. That's now scientifically proven. But if we compare them, here we see that, here we see the workloads, so you see that male mice work harder. We see a shift in plasma corticosterone, again, that we said before, but a lot more in the females than in the males. But we also saw that these females, they stopped their cycling. And that reminded us of something that's now called RED-S, relative energy deficiency in sport, and before it was called female athletic triad. So this is just a byproduct of these ecologically related studies. And we thought, well, maybe you can do a gene expression map over all these peripheral tissues also, how female athletic triad, or now RED-S, relative energy efficiency in sports, how that can be a health threat. And we saw many similarities. We see a reduced bone density, but we also see that the ovary changed the gene expression enormously. But the uterus has they're changing thousands of genes in genes expression. So basically the uterus is not a uterus anymore, not functionally a uterus anymore, if these female mice have to work very hard for the food. And this is something that relates to this sports deficiency in extreme sports, especially in women. In the males, we saw a big change in the kidney, and we still don't know what that means. That's something we need to follow up. So the idea is now that energy balance is changing this, what we call activity oscillator, and perhaps hormones like cortisol play a role in that. And I have to briefly skip this. We studied this in the wild, and we saw a very similar pattern in the wild. But what we saw is that if you give them, let me go faster. If you give them a full cover of the runways, I say the wild, but we studied this in enclosures. Here's the feeder, here's the hatch, and they can run to the food station, but we can give them covers of this runway. And the more cover we give them, the safer they feel. And indeed, if you reduce food, we see more nocturnality, but if you give them additional covers, then we see that the diurnality increases immediately. This is with full cover or with half cover and full cover again, which we see the diurnal activity increases if these mice feel safer. So what we think is that the negative energy balance is creating diurnality, but it's the diurnal predation risk or the perceived predation risk that is causing nocturnality. And if we play around with the amount of covers of the runway, we can flip them between nocturnal or diurnal activity. So that's the energy balance and the predation avoidance, I think, that both play a role. And of course, we could relate this to increased anxiety. And then then it's, oh, I thought, yeah, increased anxiety. And then it looks like cortical system and anxiety may play a role in where this activity is placed. Now, what we found recently in this paper that published a few months ago in Science is that my colleagues in Cambridge, John O'Neill's lab, they said, okay, well, is there a trace on the cellular level? And this is a study that's only done in fibroblasts. And if you entrain fibroblasts to a temperature cycle, this is how they entrain in the body, we see a fundamental difference between diurnal fibroblasts and nocturnal fibroblasts. And they did a gene analysis, gene expression analysis on these differences, and they found that there are a couple of genes that are involved in the Wnt pathway and the mTOR pathway are very important. And if you do an analysis on the whole phylogenetic tree of mammals, and you analyze those genes, then indeed you see that, here we have the KPG boundary again, indeed you see that you have a faster evolution of the genes involved in the Wnt pathway and the mTOR pathway. And the fundamental difference is that if you challenge those cells, those fibroblasts, what we see is that it is the nocturnal fibroblasts that have a much more flexible, a less robust entrainment to temperature than the diurnal fibroblasts. So it's not only that they're inverted, but they're also less robust, as if the circadian clock on nocturnal mammals is fundamentally plastic in nocturnal mammals. We think anxiety has an influence on this, and we said, okay, why don't we, how many minutes I have? Like 8 or so? Oh, okay, we're good. Okay, so last part of the talk, just a few slides. So we said if anxiety and cortisol hormones have an influence, not only energy balance, but also anxiety have an influence, then what happens if you give stress? What happens to the circadian organization if you just give stress? And this is something that's relevant for humans, of course. So here is the paradigm that we have a lot of experience with in the lab in Groningen. This is a so-called social defeat paradigm. We've trained BALB/C mice, big white mice, to own a territory, male mice. This only works in male mice, by the way. So they own a territory, and we introduce a C57 black 6 mouse. Now this mouse has this luciferase per2 reporter gene construct. And so we let them sniff and smell, and then they're separated for 5 minutes. Then there's physical contact for 10 minutes, and then if they start biting, we just take them out and we introduce the physical separation again. So it's a total of 20 minutes of stress for this black 6 mouse. We can do it at a certain time of day for 5 consecutive days. And here is actually, maybe I can show this movie. So here we see, it's in the dark, of course. We see the BALB/C mouse owning the territory. We take out the separation, and we see the black 6 mouse entered, and there's an immediate chasing. It's a bit more than chasing, perhaps. But you have to watch them closely, and we don't want to get mice to be hurt, so if there's really some biting involved, then we separate them. But this can go on for a couple of minutes, and then we think the stress is sufficient. The black mouse seeks for escape. So this paradigm, we can do it also in rats, but of course we revert to mice when possible. There's a Jovi publication that shows all the details if you're interested in this social defeat model. So if we do this to these mice, then indeed we see that some tissues, and this was done by Simona Otta and Xiaopeng Kong, we see that some tissues can change their phase, not all tissues, however. So especially here we see in the lung, look at the red trace, the rest are controls. In the lung we see a shift of about 9 hours in the circadian rhythms of the lung tissue. We see in the kidney we see a shift as well. And there are other tissues that are shifting that I won't show you. But we see the white adipose tissue is only shifting like 1 hour, and the liver is not shifting at all, probably because these mice are still eating at night, and the liver is entrained to the timing of food. So what it means is that different tissues shift differently. So the coordination, what we think is this internal function of the circadian clock, the coordination amongst tissues is weakened if there is a stressor, if there is a daily stressor. And if we change the timing of this stressor, we see that at the beginning of the night, when cortisol, endogenous cortisol is already high, we don't get much additional elevation. We don't see a shift of these tissues. But if we give it at the end of the night when cortisol is low, then we start seeing this shift in these tissues. And so the timing of the stressor is important. And we call this a chronic stressor, but it is at a certain time of day. And that makes you wonder. It is like you generate an internal shift work situation. And indeed, we see, it makes you wonder whether this is an unhealthy situation or not. And if I also present for medical doctors, maybe there's some medical doctors here in the audience as well, but if you ask, why, everybody agrees that stress and chronic stress is unhealthy. But if you ask why, nobody really knows. And the answer I'm getting is that, well, they have elevated cortisol, corticosterone levels. They say, well, why is that? Is that not an adaptation? Why would that be unhealthy? And we think this is the reason that it's unhealthy. You generate an internal shift work, jet lag program between your organs. And in fact, that is happening if you list, we know that's happening during shift work as well. And if you list all the health issues that are related to shift work, it's a big list. Like sleep problems, cognition, gastrointestinal problems, cardiovascular problems, metabolic problems, renal function, respiratory function, but also cancer, headache, and muscles. And so this is a pretty important and long list. And we see that there is equal amount of publications can be found where the same list can be drawn for chronic stress. And so this is, I think, a situation where we think that chronic stress is actually a circadian syndrome, like shift work is a circadian syndrome. And it's about time that we start thinking about chronic stress in that way. And maybe we also need circadian solutions to this. All right, that's the end of the talk. This is the group. I just wanted to say that we are also official advisor of the Dutch national team. So if we are winning these championships, I don't know whether we'll meet Spain, but if we are winning this championship, it's of course because of the circadian advice that we gave. Thank you very much. I can repeat the question.
Yes. Is there any questions? Is there anybody who wants to? I have a curiosity. So you're saying that all animals have clocks, right? Yes. So all animals have clocks, right? So and they're all light and trained. So I know of this salamander that lives like in a, how do you call it, a cave, right? And it doesn't have eyes. So do you think that animal also has a clock? And is it how does that work? How would that function?
Yeah, so that's a very interesting question. What's going on in areas where there's no light-dark cycle, like the deep sea or caves? Well, people have studied cave fish, and we see that in certain conditions, like the cave fish that feed on the droppings of bats in a cave, and those droppings only come in, of course, at night, then again we see those cave fish have circadian rhythms and train to the food. But if they don't have that, it's completely isolated, then we see a loss of circadian rhythms, like we see a loss of photoreception. There is probably also no photoreceptors left that can entrain the clock, and then the clock is also gone through evolution. But it takes a long time before it's gone, yeah.
Anybody else? Edgar.
Can you hear me?
Yes.
Okay, sorry.
I don't know about you, but yeah.
So it was very nice talk, and I was very happy that you mentioned the avenula, the one with the avenula, right? Specifically the medial avenula. So I had a couple of questions with that first. I don't remember you mentioned in the changes that you saw the medial avenula you saw in males and females, or it was independently of the sex? The circadian regulation.
Let me see. That was this slide, you mean? Yeah. Yeah, we're not making distinguishing. We're not distinguishing between sexes. So this is the other. Yeah, we don't have the habenula in this paper. But if you're interested in this, the data are all available. So you can go through these publications and you can check differences between habenula. Between sexes in the habenula expression. I don't have the results in my mind. I can't answer your question, but yeah, have a look. I mean, maybe that's the answer why males and females are different, yeah. Yeah.
More or less related with that, so in your paradigm of stress, do you see by chance any changes in the medial avenula apart from this?
We haven't looked.
We're gonna do that.
But do you expect that?
Yeah, because I mean, we expect because we are studying the medial avenula in the context of emotional dysregulation.
Yeah.
Specifically with
Yeah.
astrocytes and the glycan cells there. So
Yeah.
we have some difference in males, females, and something specific.
Yeah, yeah.
So I was wondering also if the stress can cause also changes in the circadian.
Yeah, well, that's what we suggest as well, that you have the emotional regulation of the habenula is just a small aspect of what it can do, but yeah, maybe it has to do something with this fear perception as well. The interesting thing is I didn't mention it in the literature list, we have a recent publication on light perception throughout the brain, projections, so not only these retinal ganglion cells that project to the SCN, but also normal ganglion cells project throughout the brain, not only even sort of visual ganglion cells, as we call them, they still project to other regions, like also the habenula receives input from the intrinsically photosensitive ganglion cells, but also from normal ganglion cells. And so we have a review on that, and we really want to follow that up, and we're good in the race to get funding for that. And our expectation is that the habenula is entrained by the clock, the circadian clock of the habenula is entrained by direct light input. That's why it can operate also independently from the SCN. Yeah.
Thank you.
Thanks. There was another question.
Mariana, I think. Hello? Does this work? Well, I mean,
hello, thanks for a nice talk. I agree with you that the problem of the chronic stress might be a circadian problem. So actually there are some studies in Cushing patients in which they inhibit the the cortisol production in the evening, and they do see that they improve the symptoms after. So, and in your paradigm, it seems that it's only shifting the clocks in periphery when you give the social defeat late in the dark phase. So why do you think all the peripheral tissues respond differently? So because one can think, okay, they get disorganized, and this is why all the symptoms is all these many different systems and tissues happen.
Yeah.
But why would that be? What's your
Why
idea?
different? Yeah.
Why the difference?
Right. Well, I mean, one of the obvious answers, the first answer would be, for instance, we don't see a shift. I didn't express this explicitly, but we don't see a shift. Let me see. In the top graph left, we do not ever see a shift in the suprachiasmatic nucleus. And we know why that is the case, because the suprachiasmatic nucleus is not expressing any glucocorticoid receptors. And so the obvious thing is the first level of explanation would be there may be a difference in receptor expression in these tissues. And then, of course, if that's not different, we tried to check it, and it didn't pan out for all the tissues. But yeah, I think we really need to measure it. But then, of course, there can be secondary pathways, etc. So, yeah, that's the easy answer. Part of the answer, at least.
Probably the right one.
Yeah.
Thank you.
Hi, a very interesting talk, thank you. So you focus on negative energy balance. So what happens at the other end of the spectrum with all the epidemic on obesity going on? And do you expect opposite changes in positive energy balance in obesity, for example?
Oh, very nice. Yeah, that's a very nice question. I didn't put this in the talk. So there was the hypothesis that it's just the reward, right? And it's not the negative energy balance, it's just the reward of food, and that's what's changing these patterns. So we tested this by letting them work for chocolate. Again, it's a very important paper, I think, but it's very badly cited, I have to say. But working for chocolate, you know, it's like an interesting... But we saw no effect. So they're not shifting, they love the chocolate, they prefer it above normal food, they work a lot for the chocolate, so they're willing to work a lot. All that is happening, but they don't shift their circadian rhythms. So we really think it is the negative energy balance. Yes. Yeah.
Just to add, but like when I saw the voles, if they have ad-lib food, they are like how do you active during the day? But then if you restrict their food, they become ultra diet. So
yeah.
That could be maybe related to this, no? Or like
yeah, we haven't checked that with chocolate. I don't even know whether voles like chocolate. I think.
I think they do. Yes.
But I have to say, so we don't see a shift. But if we let them work for chocolate, we also don't see a loss of weight, right? So again, it's negative. Now your question was, rethinking your question, is what happens if you have a positive energy balance?
Right.
So in humans, actually, I don't know whether you know the work of Till Runeberg, but Till Runeberg studied the sleep timing in humans. And many years ago, when he started studying this, I asked him, are you in your questionnaires, are you also asking these people for their BMIs? Because I think there's a relationship with BMIs because of our mouse results. And he said there's nothing in there. But it actually turns out that humans who are overweight, you have to somehow be overweight category, then you see a positive correlation with BMI and sleep timing. But it's the other way around. So it's basically saying that the more fat you are, the more heavy you are, the more nocturnal you become with your activity. So yes, there are indications that in that very extreme class of people, that humans with more positive energy balance become more nocturnal active. The thing is that everybody interprets this as the sleep pattern is disturbed, and that results in overweight, right? And I'm thinking, because of our data, maybe it's the other way around. Maybe these people are overeating, and that causes them to become more nocturnal. Yeah, it's a very interesting question. Thank you.
Yes, maybe the final question, because we have to go drink coffee. I think this is it, right? I don't see it very well. Okay, so the coffee is in front of this room as well as the lunch. And I think that everybody already knows that the posters are upstairs. I just want to ask the presenters of the next session to come here and put their presentations on the computer so we can be flexible.
Thank you.